about personal genomes, it is often tempting to envision those personal genomes as this logical thread of destiny along which mutations are scattered, and if you could only read those lists of mutations, you would be able to foretell at least some of your clinical future. Now, obviously, this is an oversimplification, and the reality is much more messy, and there are at least two hurdles when you look along these lists of variants that discover in personal genomes. One such hurdle is really technological. Despite the great improvement in throughput and accuracy of whole genome sequencing platforms, there are still a few errors per million in those genomes, and these pile up depending on the specific technology and pipeline to potentially tens of thousands of variants reported in every personal genome. Variants that are not really there, so just purely technical errors that you'd be concerned about when looking at your patient or the other issue is as troubling, which because these are variants that are truly there in your genome, these are so-called biological false positives. These are variants that are not de novo, you inherited them from your parents, but they are unknown, they're novel, they're not present in any of the existing databases, and it's hard to determine whether you should be worried about them because they're really damaging, or they're just not there in databases because of the incompleteness of the moving from personal genomes to the population level. Key idea is that in a population we're not really unrelated. So the Harry's genome and my genome are not really genetically unrelated, but rather they are mosaics that are sampled from a finite and often small pool of ancestral chromosomes. And more often than not, Harry and I may be lucky and sample the same ancestral chromosome in a particular region of the genome. And in this case, we share this segment that we practically identical. The way genetics use this identity of shared segments is by leveraging genotyping of large cohorts to identify those shared segments. Then, if you want to sequence this individual, you'll be able to compute the sequences of all these shared segments across all the remote hidden relatives of that sequence individual. So, in the talk, I'm going to discuss these aspects of both personalized genomics in terms of technical false positives and inherited novel variants, as well as our ability or improved ability to impute variants based on a population-specific kind of genomes. So other speakers have alluded to the unique history of Ashkenazi Jews. Our group and others have quantified this history as going through a very severe population bottleneck in the early second millennium, and that bottleneck was severe enough to limit the gene pool effectively to about 300 individuals that had expanded to the population size of millions that we have today in the Ashkenazi population. And this is also evident when you try to estimate how many such individuals do you need to sequence, 50, 100, 150, in order to cover 20, 40, 60 more percent of the population genome by these segments that are shared between the sequence cohort and individuals that you haven't sequenced. So this led us several years back to go ahead and start sequencing whole genomes. The first phase of this study was published 2 years ago in and in this data set, we published 158 healthy Ashkenazi individuals sequenced using the complete genomics sequencing platform. And by we published, I really mean Shai Carlin, who's the first author, and I think every talk here acknowledges him in some way or another. And what I'm going to spend most of the time now is the newer data, which is fivefold bigger, and is using the Illumina sequencing platform that's much more common, and that's using most cases as well as control, but are still, and these samples are still useful for people as reference samples. And this is a joint effort. It wouldn't have been possible without the collaboration across multiple institutions, both in Israel as well as in the New York area, and multiple individuals. Now, looking at such a large data set across the entire genome, we have the benefit of addressing the quality and accuracy issue head on. And the first thing that you see when you vary the quality threshold for calling particular variants, and of course the stricter you get, the less error you would have, but also the fewer true variants. And in regions that are labeled as strictly called, as well as in regions that are labeled as only pilot level called, you have a precipitous drop in error rate at a particular threshold that is helpful for us to go forward and set as the calling threshold for our data. Now, it's important and often underappreciated to realize that different regions may be either very well called or very poorly called, depending on this kind of annotation that the 1,000 genomes have. This kind of annotation that the 1,000 genomes have defined of masked regions versus pilot regions versus strict regions. It can be very well, at least when the genome is normal rather than repetitive. In low complexity, in regions that aren't as low complexity, that is regions of repeats, you suffer into quality in all kinds of regions. We took advantage of having access to multiple platforms, sequencing both duplicates as well as different cohorts, and we're able to evaluate the frequency of each variant represented by the different dots here, based on the genomics cohort and the Illumina cohort. And as you might expect from the data, the frequencies are very correlated. The vast majority of variants of non-negligible frequency are really along the Y-X diagonal, but there are a few outliers that are problematic in at least one of those platforms. Importantly, we see those horizontal line in this scatter plot at sort of round frequencies, exactly half, exactly quarter in the Illumina data, suggesting that these are kind of weird artifacts, potentially structural variants that are still called at very precise simple fashion. So moving from the technical false positive to the variants that are really there, variants that are labeled as novel based on the current version of EVSNIP, the median Ashkenazi genome in our data, a little over 40,000 variants that are novel based on last week's EVSNIP. But if you filter out variants that have been observed elsewhere in our panel, you're left with more than an order of magnitude fewer variants, a little less than it's a little over 3,000 variants per genome. Now, many address variant or filtering of novel variants for importance, not just based on whether they are observed elsewhere, but also by their functional location. Indeed, for variants along exomes, there are much larger, much deeper data sets. So even before our study, the median Ashkenazi genome only had 27 novel variants that are either coding or splicing. But still, if you also filter based on the other Ashkenazi samples, you're able to get the median sample to single digits. So that may be approaching the level of clinical utility that you might want to have. So moving on to from the more diagnostic or clinical use of personal genomes to the more research side, where you want to impute a large, often research cohort based on the reference panel. We try to impute Ashkenazi samples both based on cosmopolitan panels like the 1,000 genomes, as well as by our own Ashkenazi sequencing data, or by a combined panel of the Ashkenazi Jews sequenced and the 1,000 genomes. And listed here are error rates in imputation, so lower is better, and the panels that involve the Ashkenazi samples perform much better, both for rare allele, reference allele, homozygous, reference alternate allele, heterozygous, as well as alternate homozygotes. So this is what you probably should be doing if you impute on your own computers. An often attractive paradigm for people who don't want to do that on their own is to use an imputation server that many of the large projects are offering. Specifically, there are options to do that with the 1,000 genomes or with the UK 10K project, and also with the Haplotype Reference Consortium, which is interesting because we've actually contributed the phase 1 data already to the Haplotype Reference Consortium, which may explain why their server performs better than the other servers on Ashkenazi sequence data. Parsing these accuracy data along the frequency axis, these are so the differences between imputation based on a cohort that includes Ashkenazi samples in a large cohort, or the Haplotype Reference Consortium that includes some Ashkenazi samples versus cosmopolitan reference panels, are visible throughout the allele frequency spectrum, but are more meaningful when you're concerned with alleles that are slightly more rare, because then the quality is not as good throughout. So the small gaps are much wider. An anecdote that we can observe when we zoom in, basically with the same plot, but only looking at chromosome 20, so this is really the same non-reference allele frequency axis, and we plot the imputation accuracy of the imputation based on the Ashkenazi sequences in red, which is better than imputation without the Ashkenazi sequences in blue or green. An anecdote that shows up is here at exactly 38% allele frequency. We see that the non-Ashkenazi panels miss a lot of Ashkenazi variants. So we try to figure out what's going on, how come this is happening. And obviously this is some kind of an artifact because they are just missing a single variant, but a structural variant that's present in the Ashkenazi population. So this is the genome axis plotted, and we're plotting several of the Ashkenazi samples where we have lots of variants that are the colorful dots here that are reported within relatively short segment of chromosome 20. So there's actually, so it's not a standard pipeline, but some groups have advocated aligning the genomes, aligning not just to a single reference genome, but also aligning to a decoy genome that includes alternative versions of high complexity regions, and using that we're able to clear a lot of those issues. So it's helpful to be on top of the nitty-gritty details of your pipeline in order to avoid such potential issues. So just to wrap up, for personalized medicine, having the large cohort allowed us to improve quality control thresholds and to reduce the number of false positive variants that are reported by default pipeline, and the population specificity of this panel allowed us to remove from consideration variants that are not in databases just because the population is underrepresented in those databases, rather than because of these variants being worrisome. This allows us to have only single-digit number of functional variants per genome, so making some strides towards clinical utility. In terms of imputation, Cosmopolitan panels are rapidly expanding and are doing better and better, but obviously you'd like to work in one with a Cosmopolitan panel that also includes samples from the population you're working with, otherwise you suffer in terms of quality. As I mentioned, this is really joint work by many people. I'd like to specifically mention the group at the New York Genome Center who actually did the sequencing of the Illumina samples, as well as Cameron Palmer in my lab, Todd Lentz and Jean Yu from Long Island Jewish Medical Center that Harry mentioned, and as well as Shai Farmer, of course. Thank you.
The imputation relates to the representation of IBD segments across the genome, and how do these, both short IBD and long IBD among Ashkenazi Jews, how did these correlate?
So I think as Nora Rosenberg mentioned, in terms of long IBD segments, the Ashkenazi population does not stand out that much, and so really we are really thinking of the short IBD segments, shorter shared segments that are due to the demography of a small population bottleneck, and in terms of those explaining the improvement in imputation, it's actually nice to observe imputation methods that are based specifically on such shared segments that are coming up, and these typically require a very large number of reference samples, say in the UK, but in Ashkenazi Jews we see these methods perform very well, even with a population reference of only a few hundreds.
So 70% is both very good and also not very good. So in order to increase the accuracy of imputation, say to the level of 0.5 or 1%, how large would the sample size be?
So in order to get the imputation where we're at, we're using the sort of short segments of identified descent that are shared with individuals, but also segments that are even shorter than that we're not confidently calling even, but still the probabilistic imputation machinery is able to leverage those and sort of extend other places in the genome where we have longer segments and and get highly accurate imputation. All those comparisons between panels have been conducted using the same underlying imputation machinery for comparison. So that kind of variable factored out of the equation. Sure. So in terms of clinical significance, so that depends on the particular collaborator who contributed those particular mutations. So the particular samples that are carriers of those mutations. So this current cohort includes both cases as well as controls. So obviously, if potentially functional mutations had been found in Crohn's disease patients, then the Crohn's group is going to follow up, but definitely plans to do that. If they've been found in schizophrenia patients, they're going to follow up. Does this make sense?